Nitrogenous bases, they are the major part of nucleotides. So, what are those nucleotides? They are the building blocks of nucleic acid. Building blocks of nucleic acid. Nucleic acid means there are mainly two types of nucleic acid, DNA and RNA. So basically both DNA and RNA, they are made up of nucleotides. The major difference is DNA is made up of deoxyribonucleotides and RNA is made up of ribonucleotides. So I'll tell you what is this deoxyribonucleotides and ribonucleotides. Before that, the general structure of a nucleotide. A nucleotide is made up of mainly three components: sugar residue, phosphate group and nitrogenous bases. The sugar residues are of mainly two types: deoxyribo sugar and ribose sugar. Deoxyribose sugar, as the name indicates, which is present in DNA. Ribose sugar is present in RNA. That is why they are named deoxyribonucleotides in DNA, because in DNA the nucleotides are made up of deoxyribose sugar, and in RNA it is known as ribonucleotides because they contain ribose sugar in it. Then phosphate group and nitrogenous bases will be present. So these nitrogenous bases are of mainly two broad groups: purines and pyrimidines. Basically, the structural difference is there between purines and pyrimidines, and there are mainly two types of purines: adenine and guanine. Pyrimidines are of mainly three types: thymine, cytosine and uracil, in which adenine, guanine, thymine and cytosine, thus nitrogenous bases, are present in DNA and helps in the formation of nucleotides. In case of RNA, thymine is replaced by uracil. So in RNA, the nucleotides are made up of nitrogenous bases like adenine, guanine, cytosine and uracil. Only very few differences are there in the structure of purines and pyrimidines. It's very easy if you know the basics, or if you know the trick behind the drawing or behind the structure of this nitrogenous bases. So let us study the structure of purines and pyrimidines. First, general structure of purines and pyrimidines. First, I am going to teach you the purines. Okay. General structure of purine is basically dicyclic. It contains a hexagonal ring and a pentagonal ring in it. So while drawing the basic structure of this purines, you should know some facts like the left side of the, first I am going to draw the hexagonal ring. For that, the left side of the ring which carry alternate carbon nitrogen residue. So here carbon, then nitrogen, then carbon comes, and then nitrogen. So this is the left side of the hexagonal ring. It contains alternate carbon nitrogen residues. In the right side, which carry only carbon residues. Okay, so the hexagonal ring is completed. Before that, in the middle carbon residues, they always carry a double bond. And in the hexagonal ring, within the hexagonal ring, this double bond usually present in alternate position. So if there is a double bond here, means the next double bond comes here and the third one comes here. So the basic hexagonal ring structure in purine is completed. So attached to the hexagonal ring, there is a pentagonal ring. So how to draw the pentagonal ring? So in pentagonal ring, carbon nitrogen alternate. So here the carbon residue is present. So the next one is nitrogen, then again carbon, then again nitrogen. Then fill the gap. Okay. And in the pentagonal ring, always between the topmost nitrogen and carbon, there is a double bond. And always this double bond is, in pentagonal ring, the double bond exists only in this position. Let us number this carbon nitrogen residues. You should, in case of this purines, you should start counting the number from here. So this is 1, 2, 3, 4, 5, 6. Then it comes here 7, 8 and 9. So the basic structure of purine is completed. So once again I will repeat. The left side of purine contain alternate carbon nitrogen residues, and the right side carry only carbon residues. And between the fifth and fourth carbon atom, there always double bond will be present, and by keeping this in mind, you can draw the alternate double bond. So it will come between 1 and 6th position and between 2nd and 3rd position. In case of pentagonal ring, the carbon nitrogen residues alternate again, and between the topmost nitrogen and carbon residues, actually the double bond is exist. This is the basic structure of purine. Keep this in mind and start drawing the two different types of purines. First adenine, then I will draw guanine. Before that, this adenine can be also named 6-amino purine. 6-amino purine, and guanine can be named as 2-amino 6-oxopurine. First, let us draw adenine structure. So, as I discussed before, first you draw the general structure of purine. Once again, I will repeat. In the left side of the purine ring carry alternate carbon nitrogen residue, and in the right side only carbon residues comes. Then draw the pentagonal ring attached to it. There carbon nitrogen alternate. Since Here it is carbon, the next residue will be nitrogen, then again carbon, then again nitrogen, and fill the bond with the existing carbon atom. So now this basic structure of purine is completed, and always this double bond exists that you can draw. And before drawing other alternative double bond, you should fix the name 6-amino purine. So start numbering the atoms. 1, 2, 3, 4, 5, 6, 7, 8, 9. Since it is 6-amino purine, to the 6th carbon atom add the amino group. Okay. Then we can start adding the alternate double bond. Before that, you should know the valency of carbon is 4 and nitrogen is 3. So start counting the valency. In case of carbon, since the valency is 4, it is already attached to 1, 2, 3 atom. So 1 is remaining. Okay, already attached to 3, so 1 is remaining. So we can draw a double bond here. Now you count 1, 2, 3 and 4. The valency is filled. Let's go to the other atom, nitrogen, the first nitrogen atom. So nitrogen valency is 3. So it is already attached to 3. 1, 2 and 3. So no need to draw double bond anywhere. Go to the second carbon atom. Its valency is 4, but now it is only 1, 2. So we can draw a hydrogen atom here. Again only 1, 2 and 3. So one more bond we have to draw. So we can put a double bond here. So check whether the bond is alternate. Yes, here there is a double bond. According to that, the double bond is correctly alternating. Okay. So we finished till this carbon atom. So go to the next nitrogen atom. Its valency is 3. It already filled 1, 2 and 3. Go to this carbon atom. Its valency is 4. 1, 2, 1, 2, 3, 4. Yes, it is filled. Go to the next carbon atom. 1, 2, 3, 4. Again it is filled. Go to the 7th nitrogen atom. Its valency is 3, but now only 1 and 2 are attached. So one more we can add. So we can put a double bond here. So correctly the double bond is alternating here and here. Go to the 8th carbon atom. Carbon valency is 4. 1, 2, 3. So one more we can draw. So we can put a hydrogen atom here. Then go to the 9th nitrogen atom. Its valency is 3. 1, 2. So one more has to be filled so that we can add a hydrogen atom here. So the structure is completed. Let's go to the guanine structure. Carbon, nitrogen, carbon, nitrogen, carbon, carbon, double bond, pentagonal ring, nitrogen, carbon, nitrogen. Okay. Start counting the position. 1, 2, 3, 4, 5, 6, 7, 8, 9. The name is 2-amino 6-oxopurine. So to the 6th carbon atom add an oxo group. Then to the second carbon atom 2 amino. So to the second carbon atom you should add an amino group. Now starts filling the valency. For this carbon atom, 6th carbon atom, the valency is already filled. 1, 2, 3, 4. So no need to change. To the first nitrogen atom, 1, 2. The valency is not filled. So we can add a hydrogen atom here. To the second carbon atom, the valency is 3 now. 1, 2, 3. So we can put a double bond here. For this nitrogen atom, third nitrogen atom, the valency is filled. 1, 2 and 3. The fourth nitrogen atom, yes, the valency is filled. 1, 2, 3, 4. Same with respect to fifth nitrogen atom. With respect to seventh nitrogen atom, valency is not filled. So we can add a double bond here. To the carbon atom, here 2, here 1. So 3 is filled. So one more. We can put a hydrogen atom here. With respect to 9th nitrogen atom, 1, 2. So one more is required. We can add a hydrogen atom here. So the structure of guanine is also finished. Pyrimidines. So starts drawing. It is monocyclic. The general structure of purine is same as pyrimidine hexagonal ring: carbon, nitrogen, carbon, nitrogen. In the left side the carbon nitrogen alternate, in the right side only carbon residues, and the double bond also alternate. That's it. This is the general structure of pyrimidids. There are mainly 3 types of pyrimidids, right? Cytosine, uracil and thymine. I'll draw the general structure of all these pyrimidines together. Now, the other name of cytosine is 2-oxo 4-amino pyrimidine. So 2-oxo. So in case of pyrimidine, have to start counting from here. 1, 2, 3, 4, 5 and 6. So in the second carbon position 1 oxo group, and in the fourth carbon position there is an amino group. Now starts filling the valency. Carbon has 4 valency. In case of fourth carbon atom, only 3 is filled. 1, 2 and 3. So we can draw a double bond here. With respect to this third nitrogen atom, the valency is full. 1, 2 and 3. With respect to second carbon atom, yes, the valency is full. 1, here 2, so 3 total, and here one more, so 4. With respect to the first nitrogen atom, the valency is not full. Here there is 1, 2, so one more we can draw, hydrogen atom. With respect to 6th and 5th carbon atom, the valency is 3 now. So with respect to this carbon atom, 1, 2 and 3. Similar with 5th carbon atom also. So we have to add one more residue here. So I am going to add hydrogen atom. So the valency is filled everywhere. the structure of cytosine is completed. Uracil. The other name of uracil is 2,4-dioxopyrimidine. Start numbering 1, 2, 3, 4, 5 and 6. So in second and in fourth carbon position oxo group comes. Here always double bond will be present. So now start counting the valency. So I am not repeating here. So the final structure will be like this. You can do it yourself. Okay. Next is thymine. The other name of thymine is 2,4-dioxo-5-methyl-pyrimidine. Just remember this common name so that you can draw the structure very easily if you know the basic structure of purines and pyrimidines. 2,4-dioxo. Start counting 1, 2, 3, 4, 5 and 6. 2,4-dioxo-5-methyl-pyrimidine. So now start balancing the valency of each atom, each carbon and nitrogen atom. So the final structure will be like this. You can also count the valency and you can ensure this. So this is all about the general structure of all the nitrogenous bases. Thank you.